All right. So I wanted to briefly introduce Gunnar Carlsson. So Gunnar Carlsson completed his PhD at Stanford in 1991. Gunnar subsequently held positions at the University of Chicago, University of California, San Diego, Princeton, and then since 1991 at Stanford. Gunnar has worked on different aspects of topology, including equivariant methods in homotopy theory, algebraic theory, and since the early 2000s, he started working on the applications of algebraic topology to the understanding of data in what is known now as topological data analysis. Gunnar has mentored more than 30 PhD students and many postdocs. I was one of those postdocs, and I'm grateful for my time at Stanford and for his mentorship. Thanks, Gunnar.
Thank you, Facundo. It was certainly great to work with you at Stanford as well. What I'd like to talk about today is the following. There's a notion of big data. It's been talked about for the last 5 to 10 years as a big problem and as a huge opportunity for society and science and industry to understand all the kinds of data that we're collecting at a much increasing rate. But I would also argue that the big data is actually a misnomer. The big is not the major problem. It is certainly a problem, but I would argue that it's one that actually we've learned to handle in some systematic and good ways, to this kind of storage and so forth. But in fact, one of the other problems, perhaps the major one in making the data useful, is its complexity. I'll talk more about what we mean by complexity in a minute, but that complexity, in fact, is actually the thing that makes data hard to understand. So let's talk about that. Size isn't necessarily the most difficult problem. Complexity is a huge obstacle. It comes in many forms. So it can come in what I would call input form. That is to say, the format of the data is complicated. Maybe it's a set of molecules, and they're given to you as atoms with positions and bond lengths. That's a complex data type, or even free text might be a complex data type. There's also what I would call structural complexity, though. So even a data set that has very simple input types, so say a spreadsheet with numerical entries, a data matrix, even that can exhibit what I would call structural complexity, and we'll need to talk about that. So that complexity, since it is an obstacle, we need to come to terms with it. We need to reduce it in some way or another, and it requires an organizing principle. So let me talk a little bit about organizing principles for studying data. And I would argue organizing principles is almost synonymous with modeling, building some kind of model for the data that you're working with. So here you see Galileo's picture of Galileo, and of course here there's a very simple model that comes out of it, a quadratic equation that describes the falling of the cannonball or off the Tower of Pisa. So that's a simple model. Gives a lot of value because you can understand and predict exactly what the trajectories are going to be. And similarly here, Kepler's laws produce another situation like this where you have algebraic answers, a simple algebraic answer. Now, the thing about this kind of modeling is that typically it works best when, in fact, the phenomenon is relatively simple. It depends in a straightforward way on a small number of variables. That's typically when it's at its most powerful. Now, so what do these models buy you, though? They buy you compression. So if you have your data set, it might be 10,000 points, but if you fit a line to it, you've replaced those 10,000 points with two numbers, the slope and the y-intercept. So that's compression. The compression is such also that it gives you understanding. When the approximation is there, you understand that one variable increases with another in a prescribed way. It allows you, therefore, to predict things. But the question is, should all models be algebraic? Is the idea of modeling synonymous with algebra and equations or not? Now, I would argue that there's a simple kind of of modeling that one can talk about, which is the notion of clustering. So it's a highly developed subject within statistics and mathematics of different ways of mimicking the human notion that we have when we look at something and we break it into connected components. Clustering is the statistician's form of that, and there are many different algorithms that attempt to mimic that. But their output is a partition of the data set, and oftentimes what one finds is that those blocks of that partition are understandable in some very simple way. You might, for example, if you're studying diabetes, you might find that patients cluster into a type 1 and type 2, or other situations, as we'll see. So this is quite powerful also. It gives you a better understanding of the data, but it definitely doesn't come out of linear regression. So again, the compression is definitely there. The number of classes is typically small. Now, what I will say is that statisticians discovered early on that there is something difficult about producing these algorithms. When you produce these partitions, they often behave in counterintuitive ways and maybe in unstable ways. And so they said, actually, we need to build some different kind of model that has a little more information to it, which is, yes, we're going to break things into partitions, but actually we're going to break it into a family of partitions which get coarser and coarser as I increase a threshold parameter. Just to give you a sense of, yes, I might have a set of partition as a model, but maybe I have a dendrogram, which this is an example of, as a model as well. Okay, so what are the problems with this? Problems with algebraic modeling, I'll put them up there. Sometimes they're too rigid to deal with complex data. Most data that we see that is useful now does not necessarily fit with a small number of variables and simple relationships. You can see here situations with discrete outputs is a problem. Oftentimes, if I try to approximate data algebraically, I get very high degree approximations where the degree of understanding I get out of the model degrades, and often also requires that I do kind of a supervised analysis with an initial hypothesis. There are also problems with clustering, though. One is that there are too many algorithms, too many ways of approximating to taking connected components. Oftentimes they involve optimizing an objective function of some kind, and what should that objective function be? It requires choices of thresholds. Oftentimes also the output is too discrete. When you actually have data where there's a continuous feature to it, a progression of some kind, a continuous phenomena, it does not work so well. And even those hierarchical methods that I showed you, which were an attempt to get around that, they also have that same problem. So what's another organizing principle? I want to tell you now, I want to propose another organizing principle for complex data, and it is this, and it's sort of a mantra. The data has shape. Shape comes out of a metric on the data set, sometimes called a dissimilarity measure. And the shape matters. The shape is actually the first thing that you want to know about your data. So it tells you the overall organization. Sometimes it gives you the insight you need right then and there. Other times you need to go at it with methods beyond the shape to get what you want. Okay, now so this comes to the notion of what I would call structural complexity. So remember I talked about complexity at the beginning. Structural complexity can occur even for data that is in spreadsheet format, with small number of rows and small number of columns. And so let me give you a sense of that and why shape is a way of getting around that. So here is data this is simple in every way. Here, a linear regression will fit this, and we will do very well when you can do that. That's exactly what you want. Of course, not all data fits along lines, as we saw. We have the cluster situation here. And let me point out that the line, or the linear regression, is approximating the data by a shape, namely a straight line, that may be the simplest of all possible shapes. But the clustering can also be viewed as approximating by a shape, namely by three discrete points. Think of it as the centroids of these clusters. So approximating by shape makes a lot of sense here, and here one might hope that all data chooses to lie along a line or break into clusters. Unfortunately, the data hasn't made that choice. The data often looks a little different. Data might look like this, where in this case it doesn't break into pieces, it doesn't fit along a straight line. Instead, the shape is kind of loopy behavior. This occurs when one is dealing with time series, periodic or recurrent behavior of some kind. Time-dependent data often fits in this situation. Now, as mathematicians, we could choose, say, actually, I found a new kind of shape here that's relevant for data. I find it's a loop. Let me build a loop detector and maybe a loop fitter and so on. But I will be a little queasy when I do that because I'm not convinced that I'm going to be done even when if I build that loop detector. I'm still going to run into other stuff like this. So here, you can see here there's a different shape, a Y shape or a flared shape. This kind of thing can occur if I'm, for example, collecting data off an airliner going across the Atlantic, where I have a normal mode of behavior, which is flying at altitude in non-turbulent conditions, and then 3 extremal modes, which would be takeoff, landing, and maybe flying in turbulent conditions at altitude. So all 4 of these say we shouldn't really be in the business of selecting a finite set of model types and then building individual things like that. We should build a modeling mechanism that can cover all these shapes all at once and can be computed easily. And so in order to get at how can I build such a model that captures these shapes, we go back almost 300 years to a recreational math problem that Leonhard Euler solved. So this is obviously, the problem itself is of no particular importance. It was, can I cross the 7 bridges going across the River Pregel in Königsberg? Can I cross them all without crossing any one of them twice? So not a very interesting problem, but the method that he introduced to solve it was, he said, look, the picture on your right there, the picture contains there's a lot of irrelevant information in there. There is, the breadth of the river, how wide is the river, how big are the islands, how long are the bridges, how deep is the river. All these things are all data about this that are, that exist, but that actually don't matter at all for the solution of the problem. The solution of the problem can be accomplished by a simple approximation to it that you can see there on the left, that network where the 2 upper and lower nodes are the riverbanks, and then the islands are the 2 middle nodes, and the bridges are just edges. And so when you look at this network, it now is very easy to go ahead and solve the problem. So, yeah. Now, so that's what I want to do with data. I want to take data sets, and I want to represent them by networks like this that represent the shape. And what I'm going to show you, first of all, how does one do that, and then show you a lot of examples of how it plays out and how it works. Okay? So the question is, how do you build networks for data sets? Well, first thing is you want to use 1 or more projections off the data set. Those projections might be statistically interesting things like centrality measures or density measures. But they might be other things. They might be particular coordinates in the description of your data. Or they might be machine learning things, if you're familiar with the notion of principal component analysis or multidimensional scaling. Those could also be used as ways of projecting. Now, what we're going to do is we're only going to use that projection for one purpose, which is to bin the data into collections, into overlapping bins. So I think you see there what we've done is we've said, look, that interval on the right, the real line, is divided up into intervals that overlap. And so what I get is now overlapping bins in the data. So you can see the upper bin is red, and then the orange is the overlap. Similarly, the green and the blue and so forth. Now, once I've done that, I'm now going to apply a clustering step. I never cluster the whole data set. That's going to lose me information. I cluster only internal to the bins. So I will create a node for each bin. Okay, and that's fine. That might look like clustering. To you, it would be what's called soft clustering because the clusters actually overlap. And that's true, but what it allows us to do is to maintain some information about the distance between the data points, because what I can do now is I can draw an edge between any pair of nodes whose collections contain a data point in common. So in this case, I'm going to draw these edges like that. And you see it here. Now I have a network that effectively recapitulates that original circle that I had, and in a simplified form. Now, I don't want to be over-optimistic here. One doesn't always, of course, get this kind of perfect reproduction of what you get. What you always get, though, is an image of the data set. And features in the image are often useful. One can understand and get back to the data and find that typically they are replicated back up in the data. Okay? So topological modeling, and let me just talk about the output. Instead of being a partition or a dendrogram or a set of equations, is instead a graph or a complex network. There are lots of things you can do with it. You can put it on the screen and visualize it. Once it's on the screen and visualized like that, you can interact with it in the way that you would in Photoshop or Illustrator, and you can select parts of it and select the parts of the data set this way. Once you do that, you can also color the data set by functions of interest. So if I have a function or a variable quantity I'm interested in the data set, each node is a collection of data points. I can take the average value of that function on that node and get a coloring, which is the description of the function on the model. That's extremely useful. Oftentimes one will find, say if one is coloring by presence of fraud, you might find several hotspots of fraud within a network. Once you have the groups like this, once I have a group, select a group, I can also produce an explanation for the group. That is to say, I can give a list of the variables that most effectively differentiate that group from the rest of the data set or from another group. And then finally, feature selection and feature analysis. I'll be talking about what I think is a very interesting example here in a minute in discussing the features. But let me show you, first of all, here's a this is a very typical kind of situation. So this is in the direction of precision medicine. So this was a data set of type 2 diabetics. So people, all of whom had been diagnosed with type 2 diabetes at Mount Sinai Hospital in New York. The data set was a rather complex one. It included both electronic medical records as well as genomic information. And so even building the similarity measure there had some complexity to it. But the outcome is the network that you see there on the left. So the overall structure here is 3 large groups connected by thin wires. Now, what it turns out is that this is actually quite an interesting finding, because I don't know about you, I often read the articles that come out that say about various diseases, oh, I found that diabetes is correlated with cancer, or oh, I found that stomach cancer and stomach ulcers are correlated, that kind of thing. And that's really interesting, but not necessarily immediately useful. What these folks found is that each of these nodes, each of these groups, is a different form of type 2 diabetes. So rather than saying, oh yes, type 2 diabetes is correlated with cancer, what we're saying is, actually there's a specific group, and I can identify which group you're in, which is heavily correlated with cancer, not just a bit. And similarly, the others, there's one of them is with neurological symptoms, and one is sort of the more standard, what one thinks of with type 2 diabetes, involving obesity and kidney issues and so forth. Another aspect, though, of precision medicine is, this is work done by a microbiologist at Stanford named David Schneider, who studies the progression of infectious disease in humans and in mice. And he studies, the variables here. He studies some genomic information and also physiological information. And what he wants to study then is to say, how do you produce a model that tells you what state you're in as far as getting well? Well, let's ask ourselves, what would that model look like? Well, I would start out in the healthy state, and now I get infected. And so I start moving along an arc in that space, the high-dimensional space of features, and I get sicker and I get sicker until a certain point, the immune system starts to kick in, and now I start to get well, and I start to move back, but along a different trajectory, because I'm not just recapitulating or reversing the getting sick part, I'm coming back in a different way. And so the model then is a loopy one. Okay? And so that's what he has found here in a number of different situations, studying malaria and flu and other things. And what it allows him to do then is to have a time-invariant way of understanding what the state is in terms of the process of getting well, getting sick and getting well. And you might say, well, can't you just encode that by using time? Two problems with that. The one is that actually the time, the speed that people move through this, or the speed that subjects move through this model, actually varies quite a bit. And in fact, it can vary, in a variable way even, move through some parts faster than normal and some parts slower than normal. But another point is that when you have a patient, you don't know when they were infected. So you don't really have access to the time variable anyway. And so that is, a powerful thing here for an argument for at least that loopy kind of model of shape in this case. And of course, it comes up in many different situations as well. Okay. So, of course, breast cancer is a very common disease. One of the ways of studying it, and studying a lot of cancer and other things, is through the so-called microarray technology. It's called genomics. And this is a data set coming from the National Cancer Institute of Holland, one of the benchmark data sets for breast cancer, 272 patients, 1,500 high-varying genes are the coordinates. So the data set, if you like, is a spreadsheet with 272 rows, 1,500 columns. And what you can see here is that there's structure to this data set. It's got kind of this Y-junction aspect that I talked about with the plane, with the jetliner. In this case, what happens is that you can see on the left there are people with near normal behavior. As I move out, then I capture more and more of the disease, and then there's a split. And the split, there's one flare going up, which is a well-known piece of the taxonomy of breast cancer, and which has very bad prognosis. It includes the so-called triple-negative tumors and so on. On the other hand, there's a flare going down to the right there, and there's a little region labeled sparse data. You probably can't see that exactly, but beyond that point, that flare has the property that every single patient in that flare survived the full length of the study. So it's kind of a perfect survival flare. It's about 8% of the patients. And what you can find here is that, yes, you see that. The way that I would operate on this data set is I would say, let me color by survival. And I would get a very high value on that flare. And then I would also ask for an explain on that feature. Let me find the explanatory genes. I would get a list of the genes. So in this case, there are some very well-known ones that are involved called estrogen receptor genes, but there are others as well. Okay, now here's a point about this. So the microarray technology is far from uniform. That is to say, there are many different kinds of it, many different vendors who build the technology, and they produce different kinds of answers. It's always vectors like this, but they might choose different genes or different gene sets or what are called pathways, on and on. And so it's very difficult to compare them. So what this shows is the upper two here are that same NKI data set, done a little differently, where we've forced the survivors and non-survivors apart. And so what you see there is you see the main body is that Y trunk, and then the non-survivors are clustered in a few clusters that are off there. So now what's interesting is that the second one, the lower level, is a different breast cancer data set. And it's done with a different technology. So there's absolutely no way of comparing coordinates here. Their coordinates have nothing in common with each other. But what you see is that the structure is very similar. The structure still has this Y sitting in there, and also it's got, in this case, it's non-relapse, it's not non-survivor, it's relapse, not non-survivor. But in any event, the clusters among those non-relapse people, that actually, as you go into the data, you see that there's actually a correspondence between the groups of non-survivors and the groups of relapse patients in the second one. So what that tells you is, or it suggests to you is, that if I'm looking at this, I say I'm starting to have some confidence that I'm seeing the structure of breast cancer itself, as opposed to breast cancer as seen by an Agilent chip or an Illumina chip or whatever the technology is. I want to use this example also to point out, I mentioned that one of the problems with clustering can be that it breaks things apart that really belong together. And that is what happens here. You see, clustering proceeds only by breaking things into pieces. And when you find that something is continuous, that there actually is a progression going on, then you will quickly break things apart. And you can see here in a dendrogram model on the left there, you can see that actually it would be hard to recover this perfect survival group, which is colored in red there, the ones that correspond to that perfect survival group. It would be hard to find those from the dendrogram. Okay. Now, this is something quite new that I'm finding very interesting, so I'd like to describe it for you. Let me just say what the problem is it's trying to address. So on all these things I've just shown you, they deal with the following situation. It's actually a data set with a small to moderate size number of rows and a large number of columns. So all these genomic data sets, 1,500, 2,000, sometimes 10,000 columns. So if we go through any ordinary graphing thing, we lose because we've got so many features. There are linear approximation methods called principal component analysis, multidimensional scaling, that can get you some kind of reasonable projections, but they often miss things. And so what I want to do is I want to try to use this kind of topological modeling as a better way to represent data sets of that type where I've got tons of features. So again, the data is given in a data matrix. So what I'm going to do is I'm going to transpose the matrix so that the features now become the rows. And now I'm going to build the topological model not on the rows, not on the patients, but instead on the features, on the sensors that I'm using to measure with. So that means the genes or the gene sets. So now, if you think about it, each data point, each patient gives a gives a function on that set of features. And so what that means is I can also regard it as a function on the topological model, because remember, every node in the topological model is a collection of data points. And so I can take the average value of that feature. So I can regard the functions, I should say the original data point, as functions on some topological space, that is to say this topological model that we've built. So what that can do for you is it can give you direct insight into high-dimensional data. So the idea is, yeah, you can't even see 4 dimensions, okay? But if we've got 20,000 dimensions, but they're all realized as functions of one kind or another on a data set, then in other words, if I have a data set that can be realized as functions on a space, then I can see them because I can color this model by the values of that function. So let's take that and see what happens. So here in the breast cancer data set again, there are 3 cohorts here. So there's the nearly normal on the left, the bad survival, cohort B, and the good survival, cohort A. And so what I'm going to do now is I'm going to produce a topological model for the features in this data set. So this has got 1,500, roughly, features, and I'm building the topological model, and now I'm going to color it by the average value for each of these cohorts. And this is what happens. So if you look there, you can see cohort A is bright blue on the bottom, there in the right-hand side. There's also an island on the left, and it's also bright red up top. If I go to cohort B, which is the bad prognosis, you'll see that it is quite dramatic what's happened. The red and the blue have totally flipped. Okay? So I've got this blue that's in the upper right, although there is a group there that's remained red. But in any event, very striking difference. And what this is telling you is, by the way, if there were a single gene that were sort of responsible for all this, you wouldn't see it in this model because it would be just a single gene. It wouldn't contribute that much. What this is saying is this is a way of capturing the fact that there's large groups of genes cooperating to do something, or cooperating either in failure or success. Now, what's interesting is if I then go to cohort C, this is one that we have had difficulty getting our hands around, understanding what's cohort C. But when you look at it, you can see that it looks, first of all, like it's a weaker version of cohort B, right? It's just not quite as red in that lower left part, not quite as blue. But notice, it's not that the intensity of the blue is less, it's just that the extent of it is less. which suggests that what happens as things get worse might be that I extend the number of genes to which something applies as opposed to their expression levels just of a fixed set getting higher and higher. So in other words, having this view of it can give you a better sense, a better way of understanding this data set. And let me just say, the fact that you can you can put this up there instantly and get this look at the data, whereas otherwise, you're looking at these long lists of genes. If I were to try to, for example, find the genes that explain survival versus non-survival, I would get a long list of genes. And, it's like doing a search on Google. You look at the top 10 in your search, but you don't bother to go mostly to the second page and certainly not to the 10th page. But there might very well be interesting information in there. And in fact, there appears to be here because you'll notice is that cohort C, although it looks weaker than cohort B, it's actually much brighter red in that island on the left, which is a curious thing. Okay. Now, here's a different example. This is from UCSD Microbiome study from Larry Smarr's lab at UCSD. So he's studying inflammatory bowel disease of various kinds. So he's studying ulcerative colitis, but also Crohn's disease, and then comes in two forms, ileal and colonic. But let's just look here. So here's a network of the features. The features, in his case, he's got 10,000 features. The features are all abundancy calculations for particular microbe types, or bacterial subtypes. And so what you see here on the lower left is the healthy patients. So this is the coloring by the healthy patients. You can see there's a bright red thing there that I've circled, and it's colored in that particular way. On the right is ulcerative colitis. And so what you can see there is I've got the ulcerative colitis, and you'll see that it looks colored very much like the healthy patients in the sense that these these lower red groups, and let me just try the mouse here. These lower groups here are recapitulated over here. Okay. However, what you see is that there's this group here, which is sorry. Oh, how do I get back? Is there a way to get back? Oh, yeah, there we go. So what you'll notice is that the one difference is that there's this group right here, which is bright red. You can't see the mouse. Okay, so then let me walk over here and see if I can do it. All right. Well, let me just talk through it. What you can see there, you can see in this picture that the only difference between the ulcerative colitis on the right and the healthy on the left is that circled red group in the ulcerative side on the right. That group is not present in the healthy. And so, thank you. Now, on the other hand, in the upper right there, what you have is one of the forms of Crohn's, and I believe this is the ileal Crohn's. And you can see that it too has some similarity with the healthy, but it differs from it in that group on the. Let me see. Oh, okay. So again, in the circled group in the healthy is simply not present in the Crohn's disease up on the right. Now, okay, so this is quite interesting. What I would say is that you'll notice that there appears to be little difference here between the ileal and the colonic Crohn's, which are the two at the upper level. And that's true when I choose this set of features, where I choose all 10,000 of them. But a way that you can make this kind of model better is to choose the features that are either the most highly varying in the data set, or perhaps are the most explanatory in distinguishing between ileal and colonic Crohn's. And when you do that, you see a different network, and they're basically the same, except there's an island where there's a bright, bright difference. So in any event, it allows you to look at this data and understand it in a way that one might not have otherwise. Okay? And here is a pancreatic cancer data set. As you know, pancreatic cancer is one of the toughest cancers to deal with. This is colored. This is a small study coming out of the TCGA Institute, the database out of UC Santa Cruz. And what you can see here, it's colored by survival time, and that in fact, there is a small group of high survivors. That is to say, here, the group is very small. The whole data set is very small. It's 150 patients, but their average, that's OS there is the number of days they'd survived is 1350, so that's 4 some odd years. All of them actually are still living, in fact, whereas the overall average in the data set is a little over a year, defined exclusively by genomics. And what's interesting here is that there's more than the group going on here. In other words, you can ask yourself, as I move from the tip of that flare inward, what happens? And what you find is that the length of survival shrinks. So in other words, it's not just that we've discovered a group, but we actually can see that the structure of the graph reflects the overall length of survival. Again, survival is not part of the data. This is built exclusively out of genomic information. And so the feature space modeling for this one looks like that. Okay, and I need to get moving here. Let's see. So I wanted to talk about hotspot analysis also, because I mentioned that one can color by quantities of interest. So Lockheed is a large U.S. defense contractor and more general federal government contractor. They have thousands of projects going on at any one time with the federal government. It's very important for them to be able to determine as early as possible if one of those projects is going to be subject to downgrade. And downgrade here is just a generic term for failure of some kind. Failure to complete, complete too late, complete in the wrong way, all those things. And so Lockheed had data that consisted of, one part was kind of structured, simple information about deadlines and cash on hand and all this kind of stuff. But there was also unstructured data in the form of free text that came, in the form of the communication between people in the project and reports and so forth. So when you take all that information and build a network model on it, the topological model, it looks like this. It's kind of a simple chunk there, but the coloring here is by the actual ground truth downgrades. And so you can see that the downgrades are heavily located in that area, in the whole strip on the lower left. What was interesting is you might say, well, maybe you didn't need to go through all the trouble of doing this, the text analysis, the unstructured analysis. Maybe you could have done this just with the structured data, but no, in fact, the finding was much, much stronger precisely because of the use of the unstructured information about the text. Okay. Now, suppose that you've built by machine learning or algebraically, whatever, you've built a model, a predictive model of some kind or a scoring mechanism or something. You might ask yourself, well, maybe this scoring method is even the solution to an optimization problem. But on the other hand, maybe even though you've solved it in that way, so it's somehow optimal, but maybe nevertheless there are local areas, there are areas of systematic failure within the model that can't be reflected by the modeling mechanism because the modeling mechanism is too rigid. It's too algebraic and you can't capture these particular small groups. So this is an example of that. So this is intensive care unit data. The data actually consisted of questionnaires filled out by the personnel in the intensive care unit room concerning the patients. And so what you see here is, the upper network. So both of these are models of this data. Again, we've imposed a notion of distance on there. And the upper one is colored by the predictor that was produced by a company who was charged with building a predictor for this using genetic algorithms. And you can see that there's rather smooth motion from blue to red there. It's not an accident. It's built into the model and into the topological model. But, of course, here you actually have ground truth as well, so you can color by ground truth. And so when I do that, I see that, yeah, the model's doing something good. That far right piece, it's red, just like the model is red. But what you can also see is that in the upper left there, there's a small collection of nodes which are bright red and which in the predictor are actually green. So the predictor is saying, good to go home, but in fact these people, for the most part, did not survive. Okay? And when you go in and look for the answer to the question, it turns out to be rather simple. It turns out that for all those people in there, certain questions in the questionnaire had not been answered. So these were questions concerning the energy and the mood of the patient and so on. Who knows why they weren't answered? Maybe this even correlated. Maybe somehow things were so urgent that they couldn't fill out the entire form. Who knows? But in any event, the model doesn't work well when those kind of questions are not accounted for. So now you could improve your modeling by saying, I'm going to build two cohorts, one where those questions are answered and one where they're not, build models separately and so on. There are various ways one could approach it, but understanding the faults in your model is absolutely fundamental because all modeling mechanisms have some degree of, are going to miss some things, and you'd like to try to find the systematic ones. Okay? And just to give you a sense of it, here's another situation, mortgage default. Here's a model of mortgages for a bank. And the coloring here is by number of defaults. But another question you could ask is there a sub-borrower? Yes or no? Color by that. What is their income? Not surprisingly, high income is in that lower left blue flare where you have relatively few defaults. Where do they live? This is rural or city. You can see that city is the two blue ones below, and then the rural is in the upper one. What we have here is we have different colorings of the data set that give us insight into it. But what we can also now do is to say, let's take the bank's model, and the bank had a model already for predicting this. And let's start to color by the model error. And when you color by the model error, what you see is this. There's a red region right where the rural, or that top flare, which includes the rural folks, comes together with the urban flare. So here is a systematic area of failure. Of course, all models are going to fail on some points. If you have individual points where they fail, that's not useful to know. But if you have systematic cohorts like this, then you want to know that, and you can then correct for it. So which one can do, and then get a situation where you have a much cleaner model. Okay. And okay. So finally, I wanted to say that I've talked about some of the structures in here, loops and flares, and you may have the notion that maybe there's a systematic way of studying what are the flares and what are the loops, and you'd be right. There are ways of doing that. But a value of having a model like this is it also permits you to sometimes discover things that you didn't really expect. So this data set here is one that comes from a genetic sequence data set. It's from 3 separate populations: Han Chinese, Igbo tribesmen in Nigeria, and Caucasians in Utah in the United States. Any method that calls itself a method for data analysis will be able to pick apart these groups. So the groups here, the red one is the Chinese, the yellow is the Nigerian, and the blue is the Utah group. Okay, so if you can't fix this, if you can't find those groups, you need to go home and work on your method. But look at it here, as there's a further difference between the red group and the other two. The red group appears to be more carpet-like. It's more evenly distributed, lower degree, whereas the other two are kind of stellated and have higher degree. And so it turns out that you might hope, or you might think that this has to do somehow with the biology. Are the Han Chinese really so different from the other two groups? And the answer is no, it has nothing to do with the biology. It has everything to do with the way the data was prepared and the way the study was done. Because in this case, on the Chinese data set, there was a pre-processing step taken, which was to remove all parent-child pairs. In other words, if I had a parent and a child, I would keep only one of them. So they would remove those. Whereas that step was not taken in the other situation. We always like to think that we're doing purely science, and that it's all science. But when dealing with data, it's so often a part of the way the data is handled and prepared and structured. So to give you a sense of how serious it can be, we had another collaborator who had used this methodology on a data set he'd been working on for a long time. And he came in, and the data set, it was a database of cells and with certain structural parameters. I won't go into the details of it. But nevertheless, what he found was that there was a big group sitting on one side, disjoint from the whole rest of it, and then a major structure as well. But this big group, he looked at that and said, this is my next paper in Nature, or this is my next paper in Science. This is obviously interesting. I will have discovered something new about the taxonomy of these cells. Okay. Unfortunately, when you went in and did the explanation, found out that this was an artifact of mislabeled data. Okay. And so, it's extremely unfortunate. But nevertheless, being able to do that, being able to have a look at the data and kind of see it and get your hands on it is fundamentally important, even from the point of view of what one might call quality control and so on. Okay, now I have drastically underestimated the time that it would take me to go through this. So if you don't mind, I'm going to pass through a bunch of slides now, which I will be happy to talk to at some other point, but I need to get to the end. Yeah, this is what happens. There. Sorry about that. So I just want to sort of say, where does this fit? What does this fit? There's obviously a notion of machine learning, which attempts to understand data and so on. I would regard this as part of machine learning. It needs to be made a part of it. It needs to be used as one part of the output, one part of the way that one analyzes data sets. It's not supposed to be replace or even compete with, other methods. In fact, we use standard methods of machine learning, so-called principal component analysis, in building the networks. It's just that the key point is you want to have outputs that are more informative than just a predicted number or something. You want something that gives you more information about the data set. And so this gets into the notion of exploratory data analysis, which was developed by the famous topologist John Tukey. So you should probably laugh here because John Tukey is perhaps one of the most famous statisticians in the world. But it is true that his Princeton thesis was in topology. So we'll claim him as one of ours and say that he invented the notion of exploratory data analysis. And the point for him was to produce richer outputs that give you more information at once, that allow you to get further into the data in various ways. And so I'm going to close with a quote from him then, which I think is very much in the spirit of exploratory data analysis and the field that I would hope to contribute to with the stuff that we've talked about today. So I'll thank you for your attention and take some questions.
All right, are there any questions for Gunnar? Yes, please.
Yeah, you were using color. I was just wondering if you were considering using depth in space, using something like virtual reality and being able to do real-time simulations where you're able to actually manipulate variables and see how the shape changes in 3 dimensions?
I would love to do that. We haven't done that yet, but I know there are folks at IBM who are working hard, not on this particular thing, but a lot of kind of 3-dimensional visualization and so on, and I think there's lots that one could do there. Here, one of the interesting things about this is that what you are allowed to do is to grab parts of the network and move them around. And when you do that, it can give you some sense of the depth or the 3-dimensional structure as you rotate the 2-dimensional projection, but obviously the 3-dimensional thing would be much better.
Thanks.
Yeah.
So I have a small question. So suppose you are doing supervised learning, and you have test data. And from the test data, my understanding is that you can construct a graph from which you can learn something. But can this method like reduce the generalization error on the test data?
Right. So, yeah, well, yes, and let me say, the generalization error, which you get, you can sort of estimate or get proxies for by cross-validation. Right. So one thing that I can do is I can take the network and I can take those measures and build that as a number that I get at any point in the network. So I can actually color the network, and I might see that, yeah, I have a problem with generalization error in this part, but not in that part.
I see.
Yeah.
Yeah, thank you.
Yeah. Yeah.
Sorry.
No, go ahead.
Thanks. On your slide talking about different human populations
right.
and observing that data had been dealt with in some way and how that affected shape, we live in a time when data can often be manipulated without our knowledge.
Yes.
Is there any way to kind of reverse engineer this so that we can look and see that maybe there are some suspect structures to this data?
So what I would say is, yes, there's some hope that there's work being done now that deals with deep learning. Deep learning is one of those situations where there are sort of adversarial attacks that can be made to make them fail. And there are signs in a paper by Michael Johansson and Leo Carlsson, myself, that indicate that, in fact, yes, you can pick out the ones that are actual, spoofs or that are, adversarial.
Right. Thank you.
And let's see, this question here, there's someone who's been wanting to speak here. Maybe you can tell me your question, I can repeat it. Okay.
I'm actually going to stand up. Let me stand up.
Good.
All right, my question to you is... Sorry, I apologize. I keep on forgetting what the question is. So what I want to do is remember the question.
Okay.
So what I'm going to do to remember the question is, do you mind if I stand next to you? Is that okay?
Okay. Yeah, sure. Come on up.
Okay, the question that I have for you is, are you happy? There you go.
Yes, I'm happy.
Okay.
Yeah.
Now,
okay.
The next question I'm going to ask you is, why are you happy?
That's a long question. I mean, maybe we need to get to the question about the...
Okay. So, all right, I'm gonna skip everything that I'm gonna be thinking to get off the stage. And what I'm going to do is say thank you for everything that you've done. I really appreciate everything you've done for me because you have helped me solve the problems in my life today.
Okay, thank you. I'm glad to hear it.
All right, can I have a hug, sir?
Oh, sure.
All right, cool. Thank you. All right.
Thanks very much. Yes?
You had a lot of graphs in your presentation. So what are some common parameters that you use?
Right. So to construct them. So if you remember that slide where I showed how the circle was captured by the hexagon?
Yes.
You'll remember that there was a projection and a choice of bins in the projection. So that bin can get varied, and it can get varied in two different directions. One is by how many intervals you use, and also their degree of overlap. So we call those resolution and gain. So, you can see things at a higher or lower resolution, but also there's this gain notion that says, actually, I'm going to introduce some more edges if I increase that gain. So those are two parameters, but also, frankly, what comes out a lot is just the choice of the projections. There's a lot of knowledge that you get by working with this about what good projections are for particular kinds of data.
Thank you.
Mm-hmm. Okay.
More question there?
You mentioned in the Lockheed example that the most valuable information came from the unstructured text data. Can you say something about the shape that you found in the unstructured text data?
Well, no, you saw the shape. What I meant to say was just that using the two of them together was much more informative than using only the structured data. So what I showed you there was the network that's built off the unstructured and the structured. I don't have the network with me for this, just the pure structured.
But if it was unstructured text data, what somehow were you even looking at, I guess?
Right. So there's a whole subject of natural language processing We used very primitive parts of that, you know, where you can just get some keywords out. There's so-called TF-IDF coordinatization, which builds vectors, and then there were some particular tricks on that, but nothing fancy, nothing with diagrams or anything like that. Yeah.
All right, I think I should thank Gunnar.
Okay, thank you. Thank you.